I study seagrasses, which are marine plants that form underwater forests. Seagrasses create habitat and provide food for many organisms, including waterfowl or baby fish or oysters or crabs, and those are important for fisheries. They're also important systems because they produce oxygen, and so they are important for life on Earth. And they also provide coastal protection for us because they attenuate wave action. Our research, which is funded by Oregon Sea Grant, aims to understand how nutrient impacts that are often caused by either agricultural runoff or sewage or from upwelling, the waters that come from the deep sea, will affect seagrass beds along the Oregon coast. And we also want to know how the little grazers, the little herbivores that live on these seagrass ecosystems might help mitigate negative effects of this nutrient pollution. Nutrient pollution is one of the main causes of seagrass loss worldwide. And what happens is that these fertilizers from agriculture stimulate the growth of algae that can overgrow the seagrass and kill it. And so we're trying to understand if this is a problem in Oregon. When you have this nutrient pollution, you have this algae that grow, but you also have these little grazers, what are like little snails, sea slugs, and other animals that eat this algae. And in Oregon, we don't know which grazers are there, which these animals are there, how many are there, what are they eating, how much are they eating. So our first goal for this project is to basically understand who's there, what are they eating, and therefore if we have an increase in nutrients, can they help buffer negative effects? The results of our research can benefit multiple agencies, both at the state and the federal level, that deal with food production, fisheries, and water quality. Okay, and we have the research involves several different things. One is actually going out in the field for estuaries, so Coos, Yaquina, Netards, and Tillamook, to quantify how much seagrass there is and which animals are there. We're collecting isopods, which are one of the main grazers of seagrass, and they are like long and green. They really camouflage with the seagrass leaves. So what we've done is we're doing experiments both in the field and in the lab, where we add nutrients and we also manipulate the presence or absence of these animals to see how these combinations of more nutrients and different animals can affect the amount of algae that there is and therefore the seagrass health. And so here we have the fertilizer in a little satchel with holes, and then we also have this PVC with holes so it diffuses throughout. And here we have the same but without any fertilizer, so the satchel is empty, just to account for any potential effects that this structure could have in our buckets, like light intrusion or changes in the water flow. So we have basically this, and we'll put them in here, and then these plants will be receiving much higher levels of nutrients, simulating nutrient pollution, and then we can see how the plant responds and how the grazers also respond. Another experiment that we're setting up is an experiment to examine how these nutrients and these grazers can determine how the natives and the invasive seagrass interact together. In Oregon and in the West Coast, we have the native seagrass, which is called eelgrass, but we also have an invasive seagrass that came from Asia. This invasive seagrass is much smaller and it's faster grower. It may be better at benefiting from nutrient pollution than the slower growing native seagrass. We want to look at how one will outgrow potentially or grow better in comparison to the other, so how they compete and how that changes when we have nutrient pollution and when we have these grazers. We'll be doing feeding trials that try to see if the native animals that we have on the eelgrass beds would eat the invasive or the native seagrass, and which one they will prefer and how much would they eat of each of them. So we can understand whether there's this potential for the native animals to potentially limit some of the capacity of this invasive species to expand. And so here we have isopods that are being fed. For example, this one is fed an option of the two species. So here's the invasive and here's the native. So it can feed on both. This one just has the native species, and this one just has the invasive one, for example. It's a very large-scale project and means we have a lot of samples. Without Oregon Department of Fish and Wildlife, without EPA, and without South Slough, we would not be able to do this work. Thankfully, because of this great team of people, we're doing everything, and I think I'm very proud.